Welcome to the last video in the unit on microbial control. In this video, we're going to talk about antibiotic resistance, what that is, how it develops, and why it's such a problem in healthcare today. While we're going to focus on antibiotic resistance, I want to make sure everybody realizes that there are also antiviral resistance, where viruses are no longer being treated by their antivirals, along with antifungal resistance and antiparasitic resistance. For example, a lot of the malarial pathogens out there are becoming resistant to the antimalarials. So while you often hear about antibiotic resistance, this resistance development is happening across all the different pathogen groups that we've talked about. Let's go ahead and take a look. So when we talk about antibiotic resistance or drug resistance, what we're really talking about is a bacterial cell that has gained some sort of genetic change. That genetic change can come through spontaneous mutations or induced mutations, so some sort of mutation in the genome itself, or it can come through horizontal gene transfer where it has gained an extra gene or an extra piece of DNA. But whichever the genetic change was, it conferred a property on that bacterial cell that now it requires a higher MIC to actually stop the growth of it or to kill it. Remember that MIC is the minimum inhibitory concentration. It's the lowest amount of drug that we can use to control the microbe. If the microbe's genetics have changed, its phenotype is now different and it can handle the drug better. And so we need higher amounts of the drug in order to control it. Eventually, it's impossible to achieve those higher amounts within the body. And when that happens, we can no longer treat that organism with that drug, and we say it's become fully resistant to it. It's important to keep in mind that antibiotics are natural products. Remember, microbes have been producing antibiotics to kill off their neighbors for thousands of years. And so their neighbors have been developing mechanisms to not be killed. And this is simple natural selection. If there's something in the environment that's killing you, that something is going to select for members of the population that don't die. And so while antibiotics are always being used as natural products in the microbial populations, development of resistance to those antibiotics has also been happening for thousands of years as antibiotics have been used in this chemical warfare between microbes. And so resistance mechanisms are already out there. And this development of resistance is always going to happen. There's no way we can get around it because it's basic evolution and natural selection. So throughout the video, we're going to look at what some of those mechanisms are, but we're going to start with why antibiotics resistance is such a problem to begin with. As we look at the development of antibiotic resistance, it's important to recognize that the mutations in the genome or the genes through horizontal gene transfer are already in the population. So it's not that antibiotics are causing these mutations or these resistance genes to show up. What's happening is that antibiotics are selecting for the cells that have those mutations when the antibiotics are used. And so it's not that there's an antibiotic and the bacteria go, oh, we better mutate so that we don't die. Those mutations are already there, and this is just a case of evolution and natural selection. Something in the environment is selecting for survival of the fittest. And if there's an antibiotic there, the fittest is the bacteria that has the mutation that's able to survive. So this is what that looks like. And we have bacteria, they're not resistant, the antibiotic can kill them. Those bacteria are happily replicating and dividing. And remember, when bacteria replicate and divide, there's always a chance for spontaneous mutations to happen. And so these mutations are occurring randomly throughout the genome, and we don't necessarily know where they're happening. There's also the case for horizontal gene transfer, where one of these cells might pick up a piece of DNA that was just floating around in the environment. One or two of those mutations might actually confer resistance to those cells. And remember, this is just random. And so what happens is when you add antibiotic to the environment, that antibiotic is killing off all the neighbors that didn't have a mutation, and it's going to select for these couple that did have just a random mutation that let them survive. Because we've killed off all the neighbors, there's now a whole lot of food, there's a whole lot of space, and the bacteria that had the mutation will replicate and divide and pass that mutation now all to the daughter cells, and now we have a whole population of microbes that are antibiotic resistant. The idea being the mutation was already there in the population, And it's the presence of antibiotics now in the environment that causes the population to change and select for that mutation itself. Okay, this is an important thing to keep in mind. The mutations are already there. It's just natural selection that's occurring with antibiotic resistance. This is important because antibiotic resistance has been identified as one of the greatest threats to human health. If we look in this top graph, we can see the increase in resistance percentages for some of these. This orange line is E. coli that's resistant to certain fluoroquinolones. This pink line is E. coli that's resistant to broad-spectrum cephalosporins, one of those new groups of penicillins that we'd been trying to develop to avoid resistance. Now you'll notice there are a couple lines that go down. So this line here is going down. That's MRSA. We've done a pretty good job, especially in hospitals, of screening for MRSA and trying to decrease their prevalence rates. That's about the only one that's really decreasing to any extent. And in conjunction, we're starting to see an increase in vancomycin-resistant organisms because we've been using vancomycin more to treat the MRSA. So currently in the United States, there are about 2.8 million people that are infected every year with one of these antibiotic-resistant strains. And that results in about 35,000 deaths. And so this is a problem even in this country. We see this requiring longer treatments. So even if you're not dying from these diseases, those 2 million or so people have to be on longer treatments because we're using drugs that are not working as well. A lot of times, we have to pick drugs that now have a lower chemotherapeutic index, that lower toxicity index. What that means is they have higher side effects. The drugs themselves aren't as great, and so you feel crappy because you're taking the drug itself. And we also have a higher cost. This is costing people millions and millions of dollars for healthcare systems because of those longer durations of treatment and the higher cost of drugs. If this continues, right now predictions for the year 2050, we look at here, this is deaths are set to skyrocket. So they're predicting about 10 million deaths from drug-resistant infections when you look worldwide. And notice that shoots it over even things like cancer. So this is a problem we really do have to address because if it continues the way it is, we're expecting to see more and more antibiotic resistance pathogens, which will be leading to more and more infections that we just can't treat with antibiotics. We keep in mind that development of resistance is a natural process that's really just natural selection in action. We can see that this is always going to be a problem. Resistance is always going to be racing against evolution. And we see this when we look at the time frame between when an antibiotic has been discovered and used in treatment and when we start seeing antibiotic resistance. So for example, if you look at penicillin, remember penicillin, and you can see it on the chart there, was first discovered by Alexander Fleming in 1929, but it wasn't actually used until 1942. And if you look at the red where resistance then developed, it happened a year or two later. So we'd only been using penicillin for a couple of years when we were already seeing Staph aureus that was resistant to it. You can see the short time frames if you look at something like tetracycline, only a couple years between when we discovered the antibiotic and when we started to see resistance to it. The only one on that chart that's a little bit different is vancomycin. And that's because when vancomycin was discovered, we specifically used it sparingly. So vancomycin is not prescribed a whole lot. It's mainly only used against MRSA. And right below it, you can see methicillin and how quickly MRSA or methicillin-resistant Staph aureus was actually developed. Vancomycin, because we used it sparingly, it wasn't in the environment a whole lot, and so it took much longer to select for that resistance. Unfortunately, we are now seeing VRSA or vancomycin-resistant Staph aureus. So as we go through and talk about these drug-resistant mechanisms, keep in mind this is always going to be a race against evolution. No matter what new antibiotic we might discover, there's always going to be a resistance mechanism that the bacteria end up developing, probably only a couple years after that. Another contributor to the antibiotic resistance problem is the fact that companies have really stopped developing new antibiotics. And so most of the antibiotics that we actually currently use were discovered during what's considered the golden age of antibiotic discovery, which was really the 1940s to the 1960s. since 1980, you'll notice we actually have discovered no new classes of antibiotics. So those are antibiotics that function in a different way from some of the antibiotics we're currently seeing. And new classes are important because when you have resistance to one class, say the beta-lactamases, that resistance goes to all beta-lactamase antibiotics. So you need to really develop these new classes of antibiotics that function in a different way to try to avoid the resistance mechanisms that are already out there. And we really haven't seen this in over 30 years. And we've really been looking at needing new classes of antibiotics. The problem is, with the golden age in the 40s and 50s, some people thought, hey, infectious diseases were no longer going to be a problem. Antibiotics were the magic bullet. They were going to wipe out all infectious diseases, and we didn't have to worry about them anymore. The other problem is it's really not cost effective to companies to develop new antibiotics. It takes 10 years or so to get a new class of drug to actually being prescribed, so through clinical trials, through research and development, and that costs billions of dollars. For antibiotics, the problem right now is a new antibiotic actually won't be prescribed. Okay? It will be developed and then used and held in your back pocket for only those antibiotic-resistant infections, so that we don't develop resistance to it. But from a company standpoint, they're going to spend a lot of money developing something that then they're going to make no money on because no one's going to prescribe it and use it. And so there's this problem with development of antibiotics, even ones that are in known classes. You can see here we have a much smaller number of just new antibiotics in general. There's only been 2 since 2008. And these are ones that are in known classes. Mainly these are things like those semi-synthetic beta-lactams, carbapenems that we've been using in modifying structures of. And even those we don't have a lot of new antibiotics for. So without new antibiotics, we're really relying on trying to avoid the resistance mechanisms that are already out there for these old antibiotics that we're currently still using. When we talk about drug resistance, there's a couple of main ways that bacteria can become resistant to the drugs. And remember, that means they're increasing the amount of drug required to actually harm or control the growth of that microbe. Pathogens are able to modify the drug structure itself or degrade the drug completely. This typically requires horizontal gene transfer, and it's gaining an enzyme. And so this is a bacteria that's picked up an enzyme that's now able to fight back against the drug itself. And so that would be this classification here. And we'll look at in a minute the beta-lactams and beta-lactamase, because that's one of the classic enzymes that we talk about in this category. And so we've got enzymes that could modify drugs or degrade them, so the drug just isn't there and can't work anymore. The second way this can happen is we can change the drug's cellular target itself. So we can look at target modification. This one typically happens through mutations in the genome itself. So for example, if an antibiotic has to come in and bind to a protein of the cell to stop it from working, and you change that protein slightly so that the protein still functions but the antibiotic can't bind, now we've changed the drug's target. And by changing that target, if the drug can't bind it, it doesn't inhibit its function, and the cell can survive just fine. And then the last one is the ability to just change the concentration of the drug in itself. And that's done in 2 ways. Either through efflux pumps that take the drug and spit it out of the cell. Okay? So if the drug's getting in, we're just pumping it back out. Or through blocking the penetration itself and preventing the drug from getting in. Either way, we're lowering the amount of drug inside the cell, and if the drug concentration is lower inside the cell, it's not able to bind to all of its targets, and it's not able to do its job. These types of resistance, where we're altering the concentration, can be either through mutations in the genome of the cell itself, or they can be through horizontal gene transfer. So it could be that you have a standard efflux pump that's normally pumping out, say, waste products of the cell, and that gene picks up a mutation, and now the pump can also pump out antibiotics. The other way this could happen is you could actually gain a brand new gene that's going to prevent the drug from entering somehow, or that's going to give you a brand new efflux pump that starts pumping that antibiotic out. So as you look at the 3 mechanisms, Remember, we can change the drug structure that typically requires horizontal gene transfer to give you a gene that allows that to happen. You can change the drug's target. That's usually going to happen through mutations that alter the actual protein target of the drug a little bit. Or you can change the overall concentration in the drug. And what you'll notice is that, say, beta-lactam antibiotics are on multiple lists. They're technically on all of them. So there are multiple mechanisms to resist the same drug. And oftentimes bacteria will have developed multiple ways to do this. We tend to call those multi-drug resistant organisms because they're not just resistant to one drug, they might be resistant to many different antibiotics through many different mechanisms that they've been developing over the years. To show you a specific example, we'll look at the resistance to beta-lactam antibiotics. So remember, your beta-lactam antibiotics are things like penicillin that have this beta-lactam ring. And it's the beta-lactam ring that's important. It's allowing this antibiotic to bind to that transpeptidase enzyme and do its job. So remember, your beta-lactam antibiotics are binding to that enzyme to prevent the enzyme from cross-linking your peptidoglycan so we don't make a cell wall. Certain bacteria now have the ability to produce a beta-lactamase enzyme. And the gene for this enzyme, unfortunately, tends to be on a plasmid, and bacterial cells can pass that plasmid back and forth through either transformation or conjugation if it's an F plasmid. And that transfer back and forth is really allowing the beta-lactamase gene to spread through bacterial populations. So if a bacterial cell has the gene for this enzyme, they produce the enzyme itself, and what beta-lactamases do is they cleave the beta-lactam ring. By breaking that ring, this molecule can no longer bind. So it's no longer able to bind to its target. If it can't bind that transpeptidase enzyme, it can't stop it from functioning, and the bacteria can continue making the cell wall without a problem. So this is one of those examples of gaining an enzyme through horizontal gene transfer that now allows us to modify the drug structure so the drug no longer works. So humans play a big role in actually contributing to the development of antibiotic resistance because remember, I've stressed this a number of times, antibiotics themselves do not cause resistance. The mutations or the genes are already there in the populations, and the antibiotics are just selecting for those certain cells that have the mutation that can grow better in the antibiotic compared to their neighbors. And so what that means is that antibiotic resistance is really being selected for by antibiotics that are in the environment. The more we use antibiotics, the more we contribute to their being out in the environment in populations, the more we're going to select for these resistance mechanisms. And so humans have really caused a lot of antibiotic resistance to develop in high levels in populations because of inappropriate uses of antibiotics. And we really want to make sure we're prescribing antibiotics when we need them and appropriately, but not for things that they aren't necessarily used for. And so one thing that has been eliminated is the use of antibiotics in healthy livestock. It used to be that farmers would give low-level antibiotics to all of their animals to prevent them from getting sick. And this helped them grow better and grow bigger and get to the kind of slaughterhouse or get to produce milk sooner so they could get more money from the animals. That actually stopped in 2017. So now we can use antibiotics to treat sick animals with veterinary care, but farmers should no longer be just feeding antibiotics to all of their animals because that was putting a lot of unnecessary antibiotic out in the environment. And resistance organisms that develop, say, in cattle can very easily pass to the farmers and to their families. It can also very easily pass into the food chain, either by contaminating the fruits and vegetables or being part of the meat processing plant itself. We also have a habit and a history of using antibiotics to treat viral infections. And if you get nothing else from this whole unit, you should recognize that antibiotics do nothing against viruses. The targets of those antibiotics do not exist in viruses, and they do nothing at all to stop a viral infection. But when people have a cold or when people have the flu, they want something that's going to make them feel better, and they tend to pressure doctors to just give them a prescription, and a lot of doctors do. And so it's really important to not be prescribing antibiotics for things that they aren't going to work with, because all you're doing is putting unnecessary antibiotics in the environment, and that's causing selection of this resistance. And then the third one has to do on the patient side for people who don't fully comply with their treatments. One of the things you want to remember is if you're prescribed a 10-day course of antibiotic, you need to take it for all 10 days. If you start feeling better on day 4 and you stop and you're going to save that antibiotic in case you feel bad again in a couple weeks, well, one, nothing says when you feel bad again in a couple weeks that it's the same thing, and so you might be taking an antibiotic for, say, a cold. But two, when you don't finish the course of antibiotic, the antibiotics in your body drop below that MIC, and now you have just this low-level drug there that's able to select, because you probably haven't gotten rid of every pathogen in the body yet. As you start selecting for mutants, you start developing resistance within the body. And so it's really important that patients take their full course and that doctors only prescribe antibiotics and length of antibiotics that are appropriate to the disease we have. And we really want to limit the use of antibiotics, so we try to prevent this increased selection for resistance that we're currently seeing in the environment. So what can we do to try to treat infections if they are antibiotic resistant? So we do have what are called second-line drugs. These are other antibiotics that might have higher toxicity, right? So they have a smaller chemotherapeutic index. They might not be the best choice, but if they're better than nothing, we will use them. So there's these kind of backup drugs that we can use for resistance infections. There are also a new class of drugs that are being developed to try to inhibit the resistance itself. So for example, clavonic acid is a beta-lactam, but it's not an antibiotic. So it doesn't actually do anything against the bacteria, but what it will do is it will bind to the beta-lactamase, okay, that enzyme the bacteria was making to destroy, say, penicillin. When clavonic acid binds to the beta-lactamase, the enzyme can't work anymore, and now the penicillin can go and do its job. And so these are drugs that are not antibiotics, they're not killing the cell, but they're drugs that are trying to get around the resistance mechanism that the cell itself has developed. And this is a whole new class of drugs that people have been researching. The second thing that we talked about a little bit when we talked about viruses, and that you watched the video on for your video discussion, was phage therapy. So bacteriophages being used to lyse these cells is this new treatment option that's used in other countries, but that we're really starting to look into here, and has actually been used successfully in a couple of patients that had really severe antibiotic-resistant infections, that they couldn't find any successful antibiotic, even the second-line drugs that were working on this, and they've actually used phage in those cases with some level of success. So we're developing ways to treat antibiotic-resistant infections. The importance is going to be to recognize that every time we develop a new drug or a new antibiotic, a couple years later, there is going to be resistance that develops. So we have to keep these in mind as we do the drug development and be ready for them in the future.